Welcome back for our next session. So today we're going to be looking at our nucleic acids. So you guys probably know these better as things like our DNA and our RNA. So these are the more common names for our nucleic acid. So these we'll be investigating each of these today, focusing more on the DNA than the RNA. But we'll be looking at the individual components that make these up. So there's actually many of these nucleotides within a stretch of DNA. So each piece of DNA will be made up of hundreds of different nucleotides. So we'll see what makes those up. That's basically what we describe as the primary structure of the nucleic acid. So each nucleotide is made up of three parts. So the most important part is the nitrogen-containing base, which alternates between different bases like adenine, thymine, guanine, and cytosine. And also attached to this, we've got a sugar and then a phosphate. So when you read a sequence of DNA or a nucleic acid sequence, usually you see it represented as something like a series of these different bases, for example, ATT, AG, A. So this is just showing how the nitrogen-containing base is differing. So each one of these actually represents a nucleotide. So we'll see how we can use this code to actually work out how the DNA is going to be functional in terms of what type of protein it's going to make. We'll also look at the complementary relationship between the nitrogen-containing bases, which allows the double-stranded DNA helix to form a helical spiral. So we have one strand wrapping around a second strand of DNA to form this spiral arrangement. So it's actually because adenine can form hydrogen bonds to thymine, and also the guanine can form hydrogen bonds to the cytosine. So if you've got a guanine on one part of the one strand, and then if you've got a cytosine on the other part of the strand, we can actually get a bond forming between those two bases, and that'll actually stabilize the helical structure that we see here. So we'll look into that as well. We'll then have a look at DNA replication, and basically this is the need to copy the genetic makeup of a cell, and then we can pass that on to another cell, so passing the genetic complement of one parent cell onto two daughter cells. And then we'll have a look at our RNA and see how RNA can actually be used to convert a DNA code to a protein. So we begin with the DNA, and we then convert our DNA code to what we call a messenger RNA. And in the cytoplasm, the messenger RNA combines with our ribosome and also tRNA, and that is what's required to make our specific protein that's coded by the DNA. So we'll see how this works a little bit later on. All right, so nucleic acids, why are they so important? Basically, it's all of the information that is required for an organism to exist. So all of the processes that are hard-coded into what we are and what we do are present in our DNA. So the DNA is basically the instructions for the organism to exist the way it does. So to make all of the proteins it needs to maintain its structure, to keep things growing, to keep things in place. Basically everything that the organism does in one way or another is dictated by the genetic information. So you can think of the DNA of an organism as the stored instructions for how the organism is going to function. So the organism needs these instructions to function in the right way, basically. So we'll see that our DNA, or nucleic acids, are actually polymers of what we call nucleotides. So these are our monomers. So just like we saw in our carbohydrates lectures, where we had the monosaccharides being the monomers, and then we're building up to make the polysaccharides, this time our monomers are our nucleotides, and we're building those up to make our nucleic acids. So for example, if we have one nucleotide by itself, then we can actually attach that to another nucleotide. So what we can do is attach just more and more of these nucleotides together, and the collection of nucleotides together is what we call a nucleic acid. And remember, our nucleic acid can either be DNA or RNA. So that's what we see here. We've got our two variants of our nucleic acids. So the first part of the session is going to be focusing on DNA, and then we'll see how the RNA relates to this. Basically, the DNA's function is to store the genetic info of the organism, and if it's a eukaryotic organism, this is going to be in the nucleus within the cell. So we will need to pass on these genetic instructions, the DNA, during a cell division process. So we'll have a look at this in our DNA replication, but basically if one cell is going to become two daughter cells, then we actually need to undergo a process called DNA replication to allow us to double the DNA content in the original cell, so we can actually split that into, and both of the daughter cells can then have the appropriate DNA content. Whereas the role of ribonucleic acid, we have a few different types of these. I introduced our messenger RNA earlier. So we've got our messenger RNA, our rRNA, and also our tRNA. So we'll have a look at the roles of these a little bit later, but just briefly, the messenger RNA will allow us to take the code from the DNA and out into the cytoplasm, where it's actually required to make the protein. So the rRNA is a component of the ribosome, and then the tRNA actually brings in our amino acids to start growing the protein chain based on the code that's on the mRNA strand. So for this reason, we can say our RNAs are both found in the nucleus and also in the cytoplasm, and together they have this function of being able to synthesize protein. So it's only when we have all of these different types of RNA working together that we're able to actually create our protein from the DNA code. So we have our DNA, and within that DNA we have specific regions which we call genes, which make a particular protein, and what we'll do is we'll copy that region, and in doing this we're going to be making a mRNA, and then we start the process of taking the mRNA out into the cytoplasm, where we can start making the protein based on that mRNA code. So we do have a lot of DNA in our nucleuses. So I'm sure you've all heard of our chromosomes. So basically our chromosomes are the very condensed version of our DNA. So we can see in this picture here that what the DNA actually does is it actually winds itself around these proteins called histones, and then those histones clump together. And by looping these together, we're actually able to form our chromosomes. So there's so much DNA within our nucleus that to get it to fit in there in a compact way, we actually have to compress it. So the analogy I like to use is thinking about a piece of very long string. So our DNA is a very, very long piece of string. So how can we condense the amount of space that that long piece of string is taking up, what we can do is use these tiny little golf balls and start wrapping that string around the golf balls and then clumping those golf balls together. And that's what we're doing when we're making our chromosomes. So the string is our DNA and the golf balls are our histone proteins, and we're using these to basically condense our DNA so we can house it more easily within the nucleus. So you can see that the chromosomes are actually mostly made up of these proteins, which are our histones. So we need a lot of these to help us wrap our DNA around it. And depending on the organism, you're going to have a different number of chromosomes, and the regions within these chromosomes will also be different. So they're going to contain different genes because different organisms have different chromosomes and they're organized in different ways. For example, the human somatic cells actually have 23 pairs of chromosomes, and of these, there's actually 2 sex chromosomes. So these will be double X if you're female, otherwise you'll have XY as your sex chromosomes. So takeaway from this slide, basically that we just use our histones to condense our protein into chromosomes. And we need to do this because the amount of DNA we have is huge and we need a way of condensing this down. Alright, so here are our nucleotides. So remembering back to what we've already talked about, what is a nucleotide? Basically, a nucleotide is the smallest unit of our nucleic acid, meaning it's the smallest unit of our DNA and RNA. So nucleotides attach together to form our nucleic acids. So for that reason, we can say that our nucleic acids are polymers of nucleotides. We've got chains of these nucleotides connected together, which makes our nucleic acids. So you can think about the nucleotides as being the building blocks for our nucleic acids. And within each block, we've got these regions. We've got a nitrogen-containing base, a sugar, and a phosphate. So together, these will make one nucleotide. So the nitrogen-containing base is what we talked about before. It could be things like adenine, thymine, cytosine, or guanine. Our pentose monosaccharide is our sugar, and we'll see that these can either be a ribose sugar or a deoxyribose sugar. So this is actually what makes one of the distinctions between our DNA and RNA as well. And we've also got our phosphate group. So phosphate is actually one of our polyatomic ions, PO4 3-. So we'll be seeing phosphate quite a bit through the rest of the course, including in the metabolism lectures. So we've got a few different ways of representing this. We can show it as PO4 3-, but more often it's shown more simply as something like Pi, representing inorganic phosphate. It's inorganic because we don't have carbon attached, and you might also see it as a P with a circle around it. So any of these all mean the same thing. It's our phosphate group. Alright, so a little bit more detail now about each of the components of our nucleotide. So here we have our nitrogen-containing bases. So we saw these in one of our organic chemistry lectures. We've got both of these variants of our nitrogen-containing bases, the pyrimidines and the purines, being amines. Why are they amines? Because they contain a nitrogen connected to one or more carbons. So the R here represents our carbon chain. So where are these amine groups? I'll put some circles around them. So we've got one here, and there's actually 4 down the bottom here. So plenty of amine groups within our pyrimidine and our purine. So the key difference that I want you to take away about our pyrimidines and our purines is that our pyrimidines are actually small. So these are our single ring structures. So the way I remember this is the pyrimidine is the single ring structure that is small. So it has the larger name. So the smaller one has the larger name, whereas the purine has the shorter name and its structure is larger. So it's larger because it has two ring structures. So we call these compounds bases because they are actually slightly alkaline, and that means that they're actually good acceptors of H+. Alright, so here are our different structures of our pyrimidine and purine bases. So we can see some of the nucleotide bases that I've already been talking about with our cytosine, thymine, uracil, adenine, and guanine. So depending on whether you're in DNA or RNA, you're actually going to have a certain 4 of these. So in RNA, we have our cytosine, guanine, adenine, and also uracil. Whereas in DNA, we have same to begin with, cytosine, guanine, adenine, but instead of uracil, we actually have thymine. So that's the difference between our DNA nitrogen-containing bases and our RNA nitrogen-containing bases. Basically, in DNA, we have thymine, and instead, in RNA, we have uracil. So you can see that summarized here. Uracil is RNA only, whereas thymine is DNA only. So remember I showed you at the start that adenine pairs with thymine. So this is occurring in our DNA, whereas in our RNA, adenine actually pairs with uracil. So we have our cytosine, thymine, and uracil being our pyrimidines. So these have our smaller structures. So we can see that we've got a combination of amine and amide groups within these structures. So remember the carbonyl C double bond O with a nitrogen is actually what we call an amide. So we've got a couple of amides here in uracil. Same with our cytosine. And at the top of our cytosine here, we've actually got an amine because there's no oxygen associated with that one. Thymine actually has a very similar structure to uracil. The only difference is the extra methyl group here. So you can sort of see why it would function very similarly. And down the bottom here, we've got our larger purines, which contain a lot of amine groups. So adenine actually contains these 5 different amines, and the amine is present every time we have a nitrogen here. Whereas in guanine, there's actually 1 amide at the top here, and the rest are our amines. Alright, so the important thing to remember about our nitrogen-containing bases is which ones are actually present in DNA and which ones are in RNA. So which are our 4 nitrogen-containing bases within our DNA? So it's cytosine, guanine, adenine, and thymine. What about RNA? Which are our nitrogen-containing bases that are within our RNA? It's cytosine, guanine, adenine, and uracil. So the difference between our DNA and our RNA is that the DNA contains thymine, whereas the RNA contains the uracil. So we can see that summarized in this slide here, showing which of our nitrogen-containing bases are the purines and also which ones are the pyrimidines. And we see here also in this representation that thymine is only in the DNA, whereas the RNA only nucleotide is the uracil, whereas the other 3, adenine, guanine, and cytosine, are actually in all of the different types, so where they're in both the DNA and the RNA.